C'était déjà
J'étais médecin installé, en fait, j'ai travaillé 9 ans à l'université. À un moment donné, j'avais envie de voir autre chose. C'est comme ça qu'on m'a. Et je parce qu'il y a le seul originel et historien.
ah, c'est bon, je dois vous parler.
So I had no relevant financial disclosures. I first want to just talk about the outline of what we're going to do today. I'll first just define the cancer stem cell so that we all know what we're talking about when I describe the cancer stem cell. Also talk about why it's interesting, both for me personally and also for us as physicians at Stanford, to talk about cancer stem cells and why do we even study them. Then we'll go through the history of where we fit in, both Stanford and today, where we fit into the spectrum, what we know about cancer stem cells and what it means. Talk about some of the controversies and then some of the future directions that I think are really exciting in the field. So to start, let's define what a cancer stem cell is, and I think the easiest way to do that is to compare it to the old stochastic model of cancer, in which people traditionally thought that a single cell underwent clonal proliferation and thus led to tumors. We now understand that tumors are actually heterogeneous populations of cells, and we believe that there are certain cells that can self-renew and differentiate into transiently amplifying cells that amplify more quickly and create the bulk of the tumor. So this is primarily the difference between the traditional model and the cancer stem cell model. So why study these types of cells? Why are they important? So first of all, we talk about them all the time. Dr. Sun has talked about them, Dr. Kaplan talks about them, Dr. Lung, who's here from Hong Kong, talked about them. 3 of our past head and neck fellows have talked about them. Richard Oakley, James Tyson, Shafiq, and I talk about them a lot. They're part of Stanford history, and so when I present papers, I can actually highlight both clinicians and PhDs here at Stanford that have been significant contributors to the cancer stem cell field. And you'll see these names actually quite often: Weissman, Clark, Sunu, and Kaplan. They're also in the news. It's not just us here at Stanford and academics talking about them. This is Sergey Brin of Google. This is his wife, Anne, of 23andMe. And this is my former co-ed, Mark Zuckerberg. And they just gave the largest individual prize to a scientist. The 2013 Breakthrough Prize in Life Sciences went to Hans Kleefers for $3 million. And his work in Wnt signaling in gut stem cells, both in terms of normal physiology and cancer. But ultimately, the reason we want to talk about them is the goal, which is that we want to understand and successfully treat tumors that are resistant to our standard therapies. I thought that this diagram from the Boston Globe illustrated really well why cancer stem cells are important in that process. And that cancer stem cells are really a slowly dividing cell that then will differentiate into a progenitor cell that divides quickly. Unfortunately, most of our drugs right now, chemotherapy and radiation, target these rapidly dividing cells. And so what we're not treating is these cells that are the cell that continues the tumor or that will create tumors indefinitely. Another way to look at this is with this diagram. So let's say you have a tumor and you apply a therapy just to kill those rapidly dividing progenitor cells such as chemotherapy and radiation. It'll appear that your tumor will shrink and will go away macroscopically. But in fact, the cancer stem cell will stay there and self-renew until ultimately you have a relapse. The idea would be that if we understand cancer stem cells, we can target them. And by targeting the stem cells, the tumor may regress, perhaps more slowly, perhaps not, but ultimately will add to tumor regression and prolonged remission from disease. So in order to understand where we are and how we even got to that hypothesis, we have to go back all the way to 1953. In 1953, Nording talked about the multiple hit model in terms of cancer. And that is that a cancer results from the accumulation of DNA mutations. Now, cancer-initiating cells, even at that time, he proposed, had to survive long enough to accumulate genetic mutations necessary to generate cancer. We're going to jump to 1960, and these seemingly two unrelated fields actually need to interweave in order for this field to develop. So I think is another important point in this talk is that when interesting theories develop, it's usually a lot of people in a lot of different fields coming together and working towards an understanding of biology. In 1960, Ernest McCullough and James Till were working at the University of Toronto studying the effects of radiation after World War II. They injected bone marrow cells into irradiated mice and found that spleen colonies could arise from a single cell that they injected, and they called these cells stem cells. They also found that they could save that irradiated mouse if they injected the mouse with the stem cell. From that paper grew the evolution of the understanding of hematopoiesis, which is that you have a hematopoietic stem cell that can divide into common lymphoid and a common violent progenitor, that then can differentiate into things like B, NK, NKT, and T cells, or eosinophils, basophils, mast cells, megakaryocytes, and erythrocytes. The hematopoietic stem cell has the capability of differentiating into all of these cells, and therefore it is able to save that irradiated mouse from lethal irradiation. 16 years later, Noel hypothesized that the inherent longevity and extensive proliferative capacity of that stem cell makes it the ideal candidate for a cancer-initiating cell. Noel didn't prove this, but he at least put the two together. We've got stem cells, and we've got this cancer theory that says that you have to accumulate these mutations over a long period of time. How can cells that die over several generations do that? And these cells appear to be the perfect candidate, at least in theory. It took another 20 years before the experiment was done to show that cancer stem cells do exist. And for this, I'll take you to yet another field, and we're back to the hematopoietic field, and looking at acute myeloid leukemia. Just to remind you from medical school, AML is a rapid growth of abnormal cells derived from the myeloid lineage. What we knew at that time with the development of fluorescence-activated cell sorting, our understanding of molecular biology, which had developed over the last 20 years, was that the hematopoietic stem cell, which was discovered before, was characterized by cell surface markers. Those surface markers being CD34 high and CD30 low. When the hematopoietic stem cell differentiated into its progenitors, into its the different components, for example, the common myeloid progenitor, it obtained the CD38 marker. So we can differentiate the hematopoietic stem cell from the more differentiated cells based on its cell surface markers with flow cytometry. Now, AML is a disease that's classified by the French American British system, the FAB system. And that system is listed as the types of AML listed M0 to M7. And depending upon where you're listed in that category, describes how differentiated and what subtype of AML you have. So, what they asked, or the question that they posed, was very interesting, which is where is the stem cell in the hematopoietic hierarchy? And what they said was, let's say you've got two AMLs, you've got two patients with two different AMLs. One patient has an erythrocyte AML, that's an M6. One patient has a macrophage AML, that's an M5. If we think the cancer stem cell hypothesis is true, then we can take the most undifferentiated cell, the hematopoietic stem cell, from each of those patients, grow it in mice, and it should lead to a tumor that phenotypically resembles the patient, even though the hematopoietic stem cell does not have the markers of M6 or M5. If that hypothesis is false, then we need to take a more differentiated cell on that lineage, put it into the mouse, and that's the cell that will initiate the leukemia. And so we did exactly that. Again, I want to point out that the important factor here is CD34 high, or expression of CD34, and no expression of CD38, which is what distinguishes the hematopoietic stem cell, the least differentiated cell. So what they did is they used fluorescence-activated cell sorting. They took out the cells that were CD38 low and CD34 high, injected them into severe immunocompromised mice, and grew these tumors. And what they found was that these undifferentiated cells would differentiate into tumors that phenotypically resembled their donors, thus supporting the hypothesis of a cancer stem cell. What they also found was that if they continued to serially transplant these undifferentiated cells into one mouse after another, that they would continue to replicate, so they were able to self-renew over multiple generations. So Bonnet and Dick, and this was done at the University of Toronto, show that their hypothesis was that the leukemogenic, or for example, the tumorigenic event happened at the level of the stem cell, the hematopoietic stem cell, that then led to this differentiation of multiple tumor types, meaning that it was dependent upon the types of mutations one acquired at the stem cell level that would lead to the type of subtype of AML that you acquired, as opposed to the normal stem cell, which would differentiate into normal cells. This leads to the basic cancer stem cell theory, which is that cancer stem cells can self-renew and they can differentiate. And you will see this diagram often in a lot of cancer stem cell talks. And the reason is that we frame our understanding of cancer stem cells based on the experiments that we can do to prove that they exist, which is we take a tumor, differentiate the subpopulations of cells, we plant them into mice, and the way that we distinguish a cancer stem cell tumor is that cancer stem cell is that upon implantation, that cell can initiate a tumor and differentiate into a heterogeneous population of cells, and they can continue to do so over multiple generations. This brings us to 2003, where I'd like to point out Mike Clark, who was formerly at the University of Michigan, but is now here at Stanford. Again, names of people that are affiliated with Stanford history are going to be highlighted in red. He took a look at breast cancer tumors. So all this work was done in hematopoietic lineages. Mike Clark and Al Hodge, in their paper in PNAS, were able to take tumors from breast cancer patients. They sorted them, and they were able to implant them into mice. They identified CD44 as a potential marker of cancer stem cells. And the way they did this is they actually had a whole series of experiments where they attempted to see which cells on their effect spectrum would initiate tumors in mice. And they found that these CD44 high, CD34 negative, and lineage negative cells were the ones that did so. Not only did they initiate tumors in mice, those tumors were phenotypically similar to their donors. Those tumors, if you took the CD44 cells out of them, were able to initiate subsequent tumors in mice and continue to differentiate. Subsequent to that, multiple different solid tumors were found to have cancer stem cells. In brain in 2003, colon in 2007, pancreas in 2007, lung in 2007, ovary in 2008, prostate 2008, and melanoma in 2008. You can see that there are multiple markers that people have identified as the potential cancer. One thing that you do see, which is a fairly common theme, is CD44. So I'll pause for a moment and just talk a little bit about what CD44 is and what does it do. CD44 is a cell surface adhesion molecule involved in cell matrix interactions. It's involved in cell trafficking, lymph node homing, transmission of growth signals, uptake, and intracellular degradation of hyaluronic acid. A hyaluronic acid is a component of the extracellular matrix, and it's CD44's ligand. So because of all of these things, the thought is it may confer growth advantage. Now there are 20 exons with a bunch of variable components, so there isn't one CD44 isoform, there are multiple ones. It is preferentially expressed on epithelial cells, but it's also upregulated in activated T lymphocytes and leukocytes. So you'll recognize a couple of these names: Dr. Kaplan, Weissman, and Clark. In 2007, they took samples from head and neck cancer tumors, 3 from the oral tongue, 2 from the larynx, 2 from the floor of mouth, 1 from the oropharynx, and 1 from the maxillary sinus. And they did the proof of principle experiment that Al Hodge did in the breast cancer, which is they injected them into immunocompromised mice, and then they sorted the tumors that grew. And what you can see here is that each of these fluorescence activated cell sorted graphs is a separate patient. Here, so as you go up on the graph, these cells are CD44 positive, and these are negative. And you can see that tumors from different patients have different concentrations or different percentages of CD44 cells. implanted them into mice, they were able to show that these tumors grew out, that these cells grew out tumors that were phenotypically similar to the donor tumors. What they also were able to show is that they extracted the CD44 cells from the patient tumors, implanted them into mice, but they again produced a heterogeneous tumor, so they were able to differentiate, and they were also able to self-renew. They were able to do this over multiple generations, not just one. Thus, proving in principle cancer stem cells with the marker of CD44 positive, lineage negative. Lineage is just basically a whole list of surface markers that appear on normal cells that you exclude. So basically, if your cell expresses it, you exclude it from the CD44 population. In 2009, we say, so what? So we have these CD44 cells, and what does that mean? Well, it gets back to why do we study these cells to begin with? And what Dominic showed in 2009 was that if you take these tumors and radiate them, you enrich CD44 cells, implying that CD44 cells are the ones that are radioresistant. You can shut down and kill the bulk of the tumor cells, but you won't kill these CD44 cancer stem cells, which are supposedly slow to finish. I will mention this, I won't go into it in a lot of detail, but there have been subsequent studies that talk about other markers that can identify subpopulations of cells within CD44, such as ALDH, which is a cytosolic isoenzyme that detoxifies intracellular aldehydes. It can potentially confer chemoresistance. So there are other markers that we're discovering as we go along, but I won't get into that. 2012, we get to what's the clinical relevance of CD44. This paper, also out of Stanford, with Josh Kaplan and Weissman, took 31 patient samples from Stanford. There were 13 from the oral cavity, 8 from the lip or the skin, 4 from the oropharynx, 4 from the larynx, and 2 from the nose and the sinus. And they did the same study where they took the patient samples, they sorted the tumors to identify the percentage of CD44 that each tumor had, and they implanted them into mice to ask the question, does that patient's tumor successfully implant into a mouse or not? And what they found here on the left, this is a parametric or non-parametric coefficient correlation between two variables, is that patients that had statistical correlation between the higher percentages of CD44 and the patients presenting T-stage. Furthermore, there was a correlation between the higher percentage of CD44 and the patient's local failure. What they also did, demonstrated here on the right, was they correlated the patient's patient's ability to successfully implant a tumor with their failure, treatment failure. That included any kind of failure, local, regional, or distant. And so this was actually a great demonstration that made relevant and validated our mouse study. So we're implanting all these mice, but are all of these results simply a product of our experimental model? And what this showed is that the answer is no. This actually does seem to correlate to clinical outcomes. So this is a step further. And they did a statistical analysis of what's called the best cutoff point, which is a formula between sensitivity and specificity. And they found that if you divided the patients with tumors that had either more than 50% or less than 50%, 15% CD44 positive cells, they had a statistically significant difference between the local disease control. Further, they also had a statistically significant difference in their overall survival. Again, making a point of clinical relevance. What I thought was great about this paper was that they actually take you through 4 case models, and this I think really demonstrates the point. Take for example patient 1. It's the T2M2B-M0 oral tongue. That patient's tumor had 1.8% CD44 cells. That patient was the tumor was unsuccessfully xenografted, meaning that you could not make the tumor grow in a mouse when you attempted to transplant. The patient underwent partial gastrectomy, neck dissection, post-op radiation therapy, and 24 months later, they were disease-free. Take patient 4 that had a stage 1 T1N0M0 oral tongue, simple tumor. That patient had 73% CD44 positive cells. That patient's tumor was successfully xenografted into the mouse model. That patient had a partial placectomy and a supraormonic dissection to meet criteria for post-operative radiation. And that patient had recurrence for 6 months, even though the stage of its tumor was significantly lower than the stage of this tumor. These are 2 other corollary patients, again with high CD44 percentages, and again showing that they had failure for about 5 to 6 months. Switch gears a little bit. All of that work has been done in head and neck squamous cell carcinoma. Now I'll talk a little bit about nasopharyngeal carcinoma, which is some of the research that I participated in during the residency. So it's important to understand that, as all of you know, nasopharyngeal carcinoma is not a primary surgical disease. It's initially treated by chemotherapy, radiation, or chemoradiation therapy. And surgery is really reserved for salvage in most cases. Because of that, it's actually really difficult to obtain fresh samples. And so most of the work in NPC has been done in cell lines. And you would say that's not clinically relevant. It's been further removed from the patient. There has been a paper in 2005 by Locke that showed that cancer stem cell lines retain intrinsic stem cell hierarchies. And I'm not going to go into, again, the details of that, but there's literature supporting the use of cell lines in order to study tumors where clinical specimens are very difficult and rare. Now, the first paper was done by Wang et al., and that was done in an EBV negative cell line. So you all know there's 2 main categories of NPC, EBV positive, EBV negative. And this particular work was started off in EBV negative cell lines, which there are more. There's only a single EBV positive cell line of NPC. Excuse me. Their hypothesis was that what was called the side population on fluorescence activated cell sorting contained the cancer stem cells. And the reason they chose this side population of cells was that they selected for them by using what's called an antibody to a cell membrane transporter that effluxed the DNA binding dye. And this was by an ABCG2 mitotraxone resistance gene. And so this had been identified in other tumor types as being a marker of cancer stem cells. So they just chose this one because they were starting with nothing. They didn't know where to start. As you can see here, this particular cell line, the side population shown at the top, grows more quickly in cell culture than the non-side population. The side population is able to initiate tumors in mice, and it reflects a nasopharyngeal tumor in its histology when looked at under the microscope. What they also showed in that paper was that the side population, after 2 gray of radiation therapy, is able to proliferate more than the non-circulation cells. They also assayed chemotherapeutic agent, mitotraxone, which is the efflux, basically the drug that the cells are able to efflux, which they selected for. And again, they showed that the side population cells were able to survive longer than the non-side population cells. In 2007, Wang took this a step further and said, well, we've been studying CD44 and all these other tumor types. Why not take a look at NPC and CD44? And they did a kind of a different study, which is they used siRNA. If you guys remember from the microbiology class, siRNA is a small interfering RNA that's short and double-stranded. What it does is it unfolds, and with a complex with dicer and also RISC, it will bind to a messenger RNA and thus degrade it. So it's essentially post-transcriptional gene silencing. You have the DNA made into RNA, and then you silence the RNA before it gets made into protein. And when they knock down CD44 with siRNA in this EBV negative cell line, the tumors could no longer grow. So there is something about CD44 that's conferring a growth advantage. This cell pool is transfected with a one construct of siRNA for CD44. This is another one. This is a mock transfected pool, and this is a wild type. This is just quantified here in the graph. In 2011, there was a more formal evaluation of cell lines and their ability to form cancer, or their ability to have cancer stem cells, done by Su et al. And again, looking at an EBV negative cell line. For this, they purified by fluorescence activated cell sorting CD44 positive and negative cells. They were able to show that these cells grew faster, they had more clone formation after radiation therapy, and they survived more in a chemotherapeutic environment. But again, there was no in vivo study. There was no ability to initiate tumors in mice. So again, we're kind of evolving from that very structured and principled proof of principle experiment of what cancer stem cells were. That was initially shown by Al Hodge in breast cancer cells. So this is where we come in. Our paper, which was published in November 2012, used the C666 EBV positive cancer stem cell line. It's the only EBV positive cancer stem cell line. What we also showed was that this is a heterogeneous population of tumors. The CD44 cells can differentiate into CD44 negative cells, whereas the CD44 negative cells cannot. We implanted these two different CD40 positive versus negative cells into mice and showed that CD44 cells can initiate tumors in mice, whereas the CD44 negative, although they did grow small tumors, did not have the ability to continue and maintain tumor growth. One might ask the question, well, are you simply selecting out cells that are infected with EBV versus cells that are not infected with EBV? And what we can see here is that this looks like 0, but there's actually a low level of expression. This is a reverse transcription quantitative PCR that looks at the expression of different EBV genes, and also at oncogene. And what we showed is that basically the cells are equal in all of those parameters. So it's not a matter of whether or not they're infected with EBV or expressing EBV proteins. Now remember, fresh tumor samples, which you need in order to obtain fluorescence activated cell sorting and actually quantify a percentage of CD44 cells that exist in a patient's tumor, are really hard to come by when you're studying NBC. The best that we could do was we obtained, with the help of our pathology colleagues, an 87-patient tissue microarray that had been accumulated over multiple years here at Stanford, and we stained them for CD44. And based on that, we basically scored tumors 0 to 3, 0 being no staining, 1 being it's difficult to determine, 2 being a low-level stain, and 3 being high-level stain. And because we were able to do that statistical analysis of area of best fit based on percentage, we could only say positive versus negative. And what we saw there was, although it's not statistically significant, we do see a trend towards local recurrence and cumulative survival with the CD44 negative patients that have CD44 negative tumors as opposed to CD44 positive. This could be a reflection of the fact that it's not significant of our small n. It could also be a reflection of the fact that we couldn't quantify the percentages of CD44 positive cells within each patient's tumor, because again, we had a tissue microarray of basically tissue that had been put into a block for many years, as opposed to fresh samples that we could sort. Just want to point out that our colleagues in Hong Kong essentially published the same paper in 2012, December. And it's kind of great, number one, because we published a month earlier. Number two, they came up with the same conclusion, which is always good to hear in basic science, that another lab working independently with you comes up with a similar conclusion. And they did complement some of our findings, which is that they found in their work in C666, they were more sensitive to 5FU in their cell survival assay, and that they enriched for a particular chemokine receptor that they think may be a potential target. So that's exciting. Now there are some controversies in stem cell research. One of them comes from Sean Morrison, who used to be in my Clark's lab and is now at the University of Michigan. And what he has shown is that, or what he has argued, is that perhaps there are certain cancers that this doesn't apply to, and perhaps cancer stem cell work is simply a reflection of our experimental design, which is a difficult thing to hear if you know which one hears from us. And what he showed was, basically, if you take a melanoma cell, any melanoma cell, you make the environment permissible enough by adding in another another thing to immunocompromise these already immunocompromised mice, you can take any cell, single cell from melanoma, whether or not it's CD133, which is the marker for cancer cells in melanoma, positive or negative, and grow out the tumor. And those cells will have positive and negative cells. Another controversy that comes out of MIT, Bob Weinberg. And what he says is that cancer cells are actually plastic. They can go in and out of this cancer stem cell state in a process called epithelial to mesenchymal transition. I don't want to talk about this too much, but these are the two main arguments against cancer stem cells. Let's get to the important part, because really, the reason I was really interested in this is that to me it's one of the kind of most beautiful pathways between basic science and clinical relevance in translational research. You can really think of a way that this can help people in the somewhat near future. Let's go back to our diagram from Boston, which is why are our current treatments not working well, or what's the hypothesis of why our treatments are not working well? And our thought is that our drugs primarily target the rapidly multiplying cells. By not knowing before, not understanding cancer stem cells, we are allowing them to grow through or exist through our current therapies, and thus lead to recurrence long down the road, despite what looks like resolution of gross tumor. So cancer stem cells can explain why elimination of macroscopic disease has not led to increased overall survival in most cancers. It's a very broad statement. It's not true for every cancer. And we talked about that chemotherapy and radiation therapy target rapidly dividing cells, these bulk cells. Cancer stem cells are resistant, and they're enriched in minimal residual disease. So they may escape our general screening, and they can lead to more resistant recurrence because we know they're already resistant to things that we're treating them with. So this leads to a new model of thinking of how to treat cancer, which is that intervention with a conventional therapy will apparently create a response by taking out the bulk of the tumor, leave behind a few slow-growing cancer stem cells, and lead to a recurrence later on and resistance to therapy. By an intervention with just a cancer stem cell inhibitor, you'll knock down a few cells, so your phenotypic response will be delayed. Your tumor will look like it's just hanging out, not doing much, but eventually it will lead to a durable response. If you target both, the combination of our traditional therapy and our cancer stem cell therapy, you will get both a rapid and hopefully durable response. That's the thought. What's exciting is this new paper came out in 2013, again, with a couple of common names that you'll see here. Looking at CD47 signal regulatory protein alpha and its interaction as a therapeutic target for human solid tumors. I'm going to take you through this because it took me a while to read through this paper and kind of categorize in my mind how what this is and how it works. So, Dr. Sun, please correct me if you have any suggestions as an author on the paper. My understanding is that CD47 and siRPA are both transmembrane proteins, and when interact, they send a signal cascade that creates what's called the don't eat me signal and leads to the prevention of phagocytosis. Cancer cells have that both, and that leads to no death by phagocytosis. Normal cells only have one, maybe the other, and it leads to death by phagocytosis by these rodent cells. What this paper looked at was an antibody that blocks CD47. By blocking CD47, you block this don't eat me signal, and you allow this cancer to be destroyed by phagocytosis. Now what's really great is that they tested this. You can see how many people were involved in that paper. They tested this in multiple tumors. The diagram on the left looks at what's called a phagocytosis assay. So the more green you see, the more phagocytosis that's going on. And as you can see, when you add in anti-CD47 in this in vitro assay, you have more phagocytosis of these cancer cells. And this is true for breast, colon, glioblastoma, and bladder carcinoma. Even more interesting, they went a step further and they isolated CD44 positive cells from colon cancer. They took the cancer stem cells out and again treated them with this antibody. They showed not only do the general tumors respond, but the cancer stem cells are also phagocytosed more readily when they're treated with this antibody, thus showing that the cancer stem cells are also being affected by this novel therapeutic. We couldn't have been able to even test that if we hadn't known about cancer stem cells to begin with. They also had this great, a very clever way of understanding how much tumors are growing. They basically are able to infect a tumor with a virus that called a lentivirus that will express GFP and luciferase. Stick these mice into a bioluminescent imaging device to track the volume of tumor that's there. And what they showed is that this therapy increases the survival of mice, increases the inhibition of growth, tumor growth, and also prevents metastases. This is what the mice look like. So these are the ones treated with CD47 at 8 weeks, and here are the ones in ovarian cancer that are basically overtaking my tumor at 8 weeks. But you also see, and this is in head and neck squamous cell carcinoma, they've done assays where they showed if you inject head and neck squamous cell carcinoma cells into the tail vein of mice, they will see metastases in the lymph nodes of that mouse. If you then treat those mice with the antibody, there's a prevention of the lymph nodes. So, this is a hypothesis putting all this together, but is it possible that perhaps this anti-CD47 antibody is one of the first therapeutics that we know that combines both targeting the traditional cells as well as the cancer stem cells? Or can we potentially combine it with chemotherapy and radiation to target both groups of cells to lead to a durable and rapid response to cancers? So I also think this field is exciting because I think there's a ton of room for more research. If you do a PubMed search for, and we all know I'm going to rhinology, and how does this tie into rhinology? Well, there's a lot, Dr. Roth will tell you, there's a lot of tumors that we take out of the nose now. Search sinonasal malignancy and cancer stem cells. There are 0 articles that actually accurately match this query. The great thing about sinonasal tumors is that there are surgical diseases most of the time. So you've got lots of tumor that you can work with. One of the rate-limiting steps in NPC is that there's very little tumor to go around. And one of the great things about the Hong Kong group is that they have a collaboration amongst all of their hospitals to share and give each other tumor samples. So that's about it. That's where we are today. I think there's a lot of exciting things to be done in the future. I just want to take you back through a timeline because it really is kind of incredible how you start understanding something and how different fields weave themselves together in order to ultimately lead to a new hypothesis. In 1953, we really talked about the first multiple hit hypothesis of cancer. In 1916, Roman stem cells were discovered. In 1976, stem cells were hypothesized as an ideal candidate for a cancer-initiating cell. So in 1997, that we identified a cancer stem cell in AML. In 2003, we found it in a breast solid tumor. In 2007, we started getting into our head and neck, squamous cell carcinoma, and EBV. Again, all of this work has been done in cell lines, not patient samples, including our work here at Stanford. In 2013, now we have a potential target for at least what was proven in colon cancer. That's it. Thank you.
So go back one slide. That was a great talk. I think just want to also emphasize why basic research is the fruit of basic research may not be understood for many years. And the way CD47 was identified on cancer stem cells was by Ravi Majeti in Weissman's lab. And he looked at AML, actually. He wasn't studying breast or colon or head and neck or any solid tumor. He was studying AML. And he sorted AML stem cells versus non-stem cells, and CD47 came up as a differentially expressed gene. So that's how it came about. And then Weissman pushed for looking at it in many different tumor types, and that's how we participated, looking at it in head and neck. And it turns out that CD47 is actually widely expressed, especially in circulating hematopoietic stem cells. And the SIRP alpha that you mentioned is not expressed on the tumor cell, but it's expressed on macrophages. And it turns out that it inhibits macrophages from attacking the tumor. So the beauty of it is that it's an immunotherapeutic against the cancer stem cell. And Weissman's model for Stanford, which is unique and very interesting, is he wants to make Stanford kind of an incubator, like a biotech company. And CD47, the anti-CD47 molecule, was humanized through funds from CERM in a GMP facility type of way so that it can be put into humans. It's gone through the whole process of safety in non-human primates, and now they're writing for a phase 1 clinical trial to be done here at Stanford. So the model is, he's trying to change the paradigm where he's taking discoveries made at Stanford, applying it to disease, and then carrying it all the way through to phase 1. And then after that, it'll be licensed out, but it's all being done in-house, which is kind of interesting.